For today's webinar, we have Dr. Ed Sisko and Dr. Arun Murthy from the National Institute of Standards and Technology presenting DART-MS Data Interpretation Tool and other resources for seized drug analysis. Without further delay, here is Dr. Sisko and Dr. Murthy.
Good afternoon, everybody. My name is Ed Sisko, and I'm here with my colleague, Arun Murthy. We'll be talking about the DART-MS Data Interpretation Tool that we've been developing over the past year, as well as some of the other resources we've been developing for seized drug analysis. And so just to start off with some disclaimers, certain commercial products are identified, but that does not necessarily mean they apply endorsement or recommendation by NIST. And we need to mention that a portion of this work was funded by an NIJ award. So a real quick overview of who we are and what we do. Myself, Ed Sisko, I've been a research chemist at NIST for the last 7 years. I really focus on forensic chemistry measurement challenges, really focusing in the seized drug world, trace evidence world, and things of that nature. A lot of that is focused on ambient ionization mass spectrometry, whether that's DART mass spec or other types of ambient ionization tools, as well as a little bit of chromatography, but it's developing methods, interacting with the labs, things of that nature. Arun Murthy's been at NIST. He just hit his 5 years a few days ago. He's in the Mass Spec Data Center group at NIST, so they're the ones that can oversee the NIST GCMS data GCMS library. And he's really focused on algorithm development, searching algorithms, curation of databases, and things of that nature. But overall, we think we have two main goals as to the research we're trying to do. The first one being supporting forensic chemistry measurement disciplines by addressing measurement challenges really through collaborative research. And we found that's a key to making sure what we're trying to develop is actually of use to the community. And because of that, we hope to be able to provide forensic practitioners at all levels usable tools and resources to use at the end of the day. So real quick overview, we're going to be talking today about the DART mass spec data interpretation tool. So I'll give you a brief overview of the fundamentals of DART mass spec in case you are unfamiliar with those. So DART mass spec stands for Direct Analysis in Real-Time Mass Spectrometry. It's one of probably hundreds of ambient ionization tools, and it's one of the few that are commercially available and probably the one that's been most readily adopted by the forensics community. If you're doing conventional or traditional DART mass spec, typically what you're using is a heated stream of helium metastables to assist in sample desorption and ionization in real time in an ambient environment. And so you can see on the video on the right-hand side, the blue cylinder on the right is your DART source, and then the silver cone is the inlet to the mass spectrometer. In this instance, we're just introducing a glass rod with a little bit of sample on it in that open air stream. And we're using heated helium to desorb the analyte off of the glass rod. And then we're ionizing that using the helium metastables and a cascade of reactions that occur to ionize your sample and send that into the mass spectrometer. As you can see, this whole process is rapid. So we're talking 1 to 5 seconds of analysis time, unlike traditional GCMS type analysis where we're talking tens of minutes to do analysis. With relatively high sensitivity, and then depending on the range of mass spectrometers, you couple that DART source too, you can do a range of different types of experiments. And last year, we released a DART mass spec review article that covered forensic applications of DART over the last 5 years. So if you're interested in learning more about the fundamentals or how it's been researched and is being used by the community, feel free to check that article out. So real quick, just to set kind of the data mindset, because one of the big challenges when labs bring on DART mass spec or other ambient ionization tools into their laboratories is adjusting from the traditional GCMS mindset to how the DART data actually looks. And so in your traditional GCMS on the left-hand side here, we have your chromatograph at the top. Obviously, we're separating the peaks of interest based on chemical properties. And then when a peak pops up in our chromatogram, we can pull out a mass spec that is unique to that compound of interest. And we can go through our library and search that using a range of different tools that are available. With DART mass spec, we are no longer doing chromatography. And so that initial mass spectra is actually a mass spectra of all of the different compounds that are present in your sample. And you're seeing that simultaneously. What we can leverage with DART mass spec is called in-source CID, or in-source collision-induced dissociation. And so in the inlet of the mass spectrometer, where we're going from ambient to high vacuum, we can increase the number of collisions molecules or ions we'll see in that area of the mass spectrometer, leading to enhanced fragmentation. And so with a number of the tools that are used, the forensics labs are most commonly using a time-of-flight mass spectrometer that doesn't have a quad in front or a triple quad system or anything like that. We can use this in-source CID to obtain a typically protonated molecule spectra, and then we can go through and fragment the mass spectra to get fragment information as well about the compounds that are present in the sample. But again, even those fragment spectra, we're fragmenting everything at one time, having any sort of separation whatsoever. So I'm going to throw it over to Arun, who will talk a little bit about the data interpretation tools that are traditionally used for GCMS.
Thanks, Ed. So as Ed mentioned, the data for DART mass spec is a little bit more complicated than EI mass spec, but before we can address interpreting the DART mass spec, we kind of need to go over the tools we have existing for electron ionization mass spec. And so we have what's called the Automated Mass Spectral Deconvolution and Identification System here at NIST, or AMDIS. And what this software allows you to automate the extraction process, so pulling the mass spec of each compound from the mass spec in a consistent and reproducible manner, and then searching those mass spec against a NIST mass spectral library, or any mass spectral library, I guess, but in this format, to potentially identify those compounds. So that's a one-step process for identifying compounds via GCMS. Sometimes compounds can't be identified via AMDIS, and so what you might do is send a NIST MS search or a NIST MS interpreter, which is a comprehensive set of tools for interacting with mass spectral libraries, and it includes a variety of useful algorithms and data interpretation tools to help identify particular peaks within a mass spectrum. We also have highly specialized tools like the NIST fentanyl classifier, which allows you to interact with mass spectra of potential fentanyl animals by searching a specific library or a smaller subset library of just fentanyls, and doing additional machine learning to help attempt to localize the site of modification between fentanyl analogs. And so this is a way to identify things that aren't necessarily in your library. The common thread amongst all these tools, and so some of these tools you can use without a mass spectral library, but the common thread to effectively use all these libraries or all these tools is a reference mass spectral library. And so naturally, we figured if we want to start interpreting DART mass spectra, we should start building a DART mass spec library. There's several challenges to building a DART mass spec library, but there are two key challenges that we want to key in on for this talk. So first is the actual development process of the in-source collision-induced dissociation mass spectral library. One, there are challenges with actually procuring and measuring relevant compounds. You know, we're a big government organization, and so procuring relevant compounds while they're still relevant is not always something that we can do, but we try to be on the cutting edge in terms of buying compounds as they are released by commercial vendors. The other and more scientific challenges are determining whether the spectra we collect are actually appropriate measurements. And so normally what we would do is we would have a human, an expert, evaluate the mass spectra, because the thing about mass spectra is they can really be thought of as roughly reproducible. And so what that means is if I take a replicate measurement of a compound, I would expect that those replicate measurements will be self-similar, but I would never expect them to be identical. There will always be some baseline noise that's absolutely unavoidable. And so we'd have an expert evaluate the mass spectra and decide if that level of noise is appropriate to include in a library. And then the last thing that's really important is we need to make sure all the non-spectral data associated with that compound is correct. There's nothing worse than having a perfect measurement that's mislabeled. That makes that measurement absolutely useless to everyone. And so the last two, the scientific issues, are actually interesting issues that we can really start to address with computation. We can start to come up with algorithms and models that help us quantify potentially the noise in the mass spectrum and help us more objectively select which spectra are included in our library. And we can also automate away a lot of the tedious tasks, like making sure that the non-spectral information is correct. Molecular formula is consistent with the name, is consistent with the smiles or the structural information, and so on. So one afternoon, when we were trying to come up with a streamlined way to evaluate the ISCID mass spectra to include in the library, one afternoon, Ed comes by my office and we decide that we should come up with some numerical approaches, some algorithms and models that will help us streamline our evaluation process. One, that'll help be more objective with deciding what spectra are included in the library. And two, that'll actually really help us speed up the process and build libraries that are relevant while compounds are still relevant. So we developed a simple script with several filters and algorithms to help us assist in evaluating the mass spec. Naturally, we made the script publicly available, open source and publicly available through GitHub, and we wrote a small paper summarizing the methods. As far as that's kind of the workflow for computational research: is you solve a problem, you make the solution available, and then you write a paper summarizing it. So for people that are, and so with this script and with all the hard work collecting mass spectra over the last 24 months, we've been able to release an updated DART mass spec database of forensic compounds, currently at 895 compounds, and it's growing Pretty regularly. I was going to say quarterly, but that's not always the case. But within every 3 to 6 months, we try to include an update with relevant compounds. Each compound has 3 in-source collision-induced mass spectra, a low fragmentation, a mid fragmentation, and a high fragmentation. And there was an original DART mass spec library, and sorry, I forgot to mention that, but we've updated that. That was released in 2012, and we've updated it significantly since then, only carrying over 131 entries from that original library. And so the current update, Dragonfly, was released, I guess, late September, early October. And if you'd like to download the library, it's available in several different formats. Feel free to follow the link on the right over here. But one of the things we realized while putting together the library, and so I'm new to DART mass spec, Ed has a lot of experience with it. He pointed out to me that even if you've got a pure compound mass spectral library collected, DART mass spec, nobody uses DART mass spec for pure compound identification, which is the traditional task that electron ionization and electrospray ionization mass spectral libraries are used for. Instead, what people are using, or what the forensics community is trying to do with DART mass spec, is pre-screen. They're trying to identify several compounds in a mixture, that mixture representing seized evidence. And so our existing search algorithms for pure compound identification just weren't appropriate for the task at hand, and thus made the library less useful than it could have been. And so we sat down and we decided we should come up with a new method of leveraging our pure compound library to help forensic chemists with the tasks that they're really interested in, which is identifying components in a mixture. And so we got together again, spent an afternoon kind of, working through some ideas. Ed explained some chemistry to me, I explained some math to him. A couple hours later, we came up with the ILSA, the inverted library search algorithm. And so this method, just roughly speaking, tries to identify the library, the pure compounds that are in your library, by searching against the query mass spectrum of a mixture. And so it inverts the library search process, where you're actually treating the mixture spectrum as your search space and the library spectra as your query. It's a fun little method, and we found that in our proof of concept, it worked really well on several of our test examples. So naturally, we implemented the algorithm as a simple application and made it publicly available through GitHub, and then wrote a paper to summarize the algorithm in detail. Perfect computational research trifecta. But what we had done to that point is what we would call functional research. We addressed an interesting question an interesting scientific question. We made all of our work publicly available and summarized everything through papers and written descriptions. And so anyone in the scientific community could critically assess our work and potentially build on it. So that's really what we call functional research, the job of a scientist. The thing at NIST is that we're not just scientists, we're also public servants. And so one of the things we want to do with our research is not just entertain the intellectually curious, but also help bench level chemists that are trying to do their job. And the bench level chemist is not so impressed by our papers and our spaghetti source code, rather they need usable tools. And unfortunately, functional research does not necessarily equate to being a usable tool. But that was our goal. Our goal was to take our functional research and make it a usable tool, especially the ILSA being such a powerful algorithm. It felt like it would be a waste if it got lost in the scientific literature. And so our goal over the last several months has been to transform the ILSA into a usable tool by building a software product around it that we call the NIST NIJ DART mass spec data interpretation tool, or DIT for short. And so I should mention that Ed and I are both scientists at heart, and so we don't have a lot of experience developing software tools. In fact, we are complete novices, but we figured there's a couple of things we probably needed to get this project rolling. We needed some funding, and so we were able to leverage our existing funding at NIST as well as some additional funding from the NIJ. We needed some end users, because one of the things we decided is if you're trying to design a usable software tool, you should probably work with the people that might actually use your software. And so we were able to contact these 5 labs that we've shown here, and they were able to get involved with the software development process. And we were also able to add some expertise. Ruthie Corzo, another scientist here at NIST, as well as our prep student, Stephen Tennyson, a computer science student from UMD College Park, both of them were able to help us kind of put our vision, or work towards our vision of building this usable software tool. So now I'm just going to walk you through like the development of our data interpretation tool. We spent the first kind of few months finishing up our papers, making sure they were submitted, making sure we got good peer review to confirm that our science was solid, and also making our database available. We also spent a little bit of time implementing the ILSA functionality into the preliminary version of the DIT. This preliminary version really only having the ILSA functionality and the ability to view a library. But we shared that first version of the DIT with our user, and we also published a second update of the DART mass spec forensic database, adding another 30 or 40 compounds, I'm not sure. After sending the DIT out to the user group, a couple weeks later, we tried to collect some feedback from the user group. In particular, we used a survey that kind of outlined possible features that we could add and asked the users to prioritize these features as, completely necessary to nice to have to, completely unnecessary. You guys don't know what you're talking about in your ivory tower. So we had that out there. We also had some open-ended questions in case they wanted to add their own features or add some other opinions or share some. And with that survey, what we learned was that all the features were needed. And so the nice thing was that there was some prioritization. And so we got to work on developing the second version of the DIT based on the feedback from that. We shared that second version. And then we also simultaneously published another update. And as I mentioned earlier, we were trying to do quarterly updates of the database. And that's something we're going to continue to try to push for, but we'll see how that goes. So a couple of weeks after sharing the second version of the DIT with the user group, we got some more feedback. This time in the survey was more general, open-ended. Really, we had added a significant number of features between versions 1 and 2 of the DIT. And so what we wanted to do was make sure that the code still worked. And if it wasn't working, that it was failing gracefully. It was really less about feature addition and more about software performance. We got our feedback. It was generally very positive. We were able to share a final version of the DIT with the user group just to address any last-minute questions, make sure that everything was solid with everyone. And then we also published a fourth version of the DART mass spec forensic database, which, as I mentioned earlier, was up at 895 compounds. And so I'm actually going to pass it on now to Ed so he can actually show you how this DIT works.
Awesome. So if we pull up our DIT, when we open the DIT tool, this is basically what you get. So at the top of the DIT, we have a banner which has 3 different options. We have our search tool, which is kind of the meat and potatoes of the DIT. We have a library tab, which we'll demonstrate in a second, but that goes through and you can view all library compounds, view the metadata and all of that. And then we have the About tab, which just has all the necessary disclaimers about the software. So with the search tool, what it's meant for is in-source collision-induced association or in-source CID mass spectra generated at 1 or more fragmentation voltages. And so for the traditional setup, we started with the requirement to have 3 different voltages. Now you can upload any combination as long as you upload a low fragmentation voltage. And so on the left-hand side in this gray box is the option to load low, mid, and high fragmentation spectra. And you can upload any 1 or more of those combinations. You also have the option to choose between reference libraries. As of now, we just have our Dragonfly update. We are working on negative mode libraries, and we are continuing to update the positive mode libraries as well. On our top banner here, we have a bunch of fill-in tabs for reporting functionality. So one of the big things from the first version of our software that laboratories wanted was a way to generate a printable or PDFable report that contained all the relevant information. And so users now have the ability to type in any information up there they want: date, instrument ID, case number, item number, any other open comments they might. There's also the ability to download and upload presets. So if there's different settings, which we'll talk about in a second, or text fields that you want to always populate when you open up the software, you can download a preset file or create a preset file. And then whenever you open the software, either it'll upload in that save state, or you can load a different set of presets to change the settings around, things like that. If you have a multiple lab system, you can share that preset file across different computers or different versions of the search software, so that everybody has the same parameters when they go to do their search. Our goal of this was to keep it as simple as possible, to not overwhelm the users with a bunch of different functionalities and choices they'd have to make. And so there's only a few different types of settings that you have to set. First one is relatively straightforward: pure compound or mixture analysis. So basically, do you want to only look for one compound, the most abundant compound in your spectra, or do you want to be able to search through all the potential compounds that might be present? There's a target assumption functionality. Do you want to the way the ILSA works is we initially try to identify targets based on one of these two assumptions. So the assumption that all compounds are protonated molecules, or the assumption of using the base peak in the reference library spectra. You have the ability to only display targets where you have potential matches, which we'll show you in a second. And then the two most important ones is the identification threshold. So this is the % relative intensity threshold that you want to identify peaks above. And then your mass M/Z tolerance, which can either be integer resolution or we can set at any value between 0 and 0.1. And you also have an update search button here. That way, if you load some spectra and realize you need to change, let's say, the identification threshold, you can just hit update search and it will update that spectra for you. So it's as simple here as loading a low fragmentation volt spectra. So for instance, if we load the 30 volt spectra just to do a simple search, you can upload a spectra just like this, hit search database, and you get some identification. So in this sample, for instance, we can see we have quinine, potentially fluoxetine, amphetamine, heroin, and that's about it. Because you see here, there's a lot of peaks that aren't present in the library. What we can do in our settings tab is we can say we only want to display targets with potential matches, and now we only see the ones where we have an identification from your mass spectra. We have two different scores here, and if you, in the user manual, it kind of explains what these two different scores are, and as well as the ILSA paper, but there's an FPI score, fraction of peak intensity explained, and then your traditional reverse match factor, which is common, can be used as well in EIMS data. So in addition to identifying your targets, we have the mass spectra that are produced. You can zoom in and hover over all of this. You can get the M/Z values. Red peaks are peaks above your intensity threshold, which is your dotted line. And then you have the peak table as well, which you can choose. Do you want to see anything above the threshold, your top 10 peaks, or all peaks that are present in the spectra? You can also then upload higher intensity fragmentation voltages, and you use that to get better scoring on the search algorithm, because now we will look at not only the protonated molecules, we'll also be looking at the fragment spectra. And so you can see that when we do this, the search scores at the bottom now will change. And now you have the multiple fragmentation spectra that you can go through and look at as well. And so that's kind of what the search functionality of the DIT looks like. What we want to do, most labs now would want to go and make a report from this data. And so we can go and we can type things like today's date. We could type in an instrument ID. We could type in a case number. And then what we can do here is we can actually click the reporting button and we'll generate a report that's now in a printable or PDFable format. And so all of your metadata is now up here. Your low fragmentation 30 volt spectra is right here with the M/Z values. If you have multiple fragmentation spectra uploaded, they'll also be shown. And then at the bottom, you'll have your potential matches as well now. So target 1 with quinine, target 5 with amphetamine, and target 7 with heroin. So that basically is how the search tool works. It's really that simple. And then there's also a clear search button here where we can eliminate everything and just start from scratch all over again. And then we also have download presets and load preset op functionality as well. Our library tab allows us to go through and actually look at different library spectra. Let's say, for instance, we wanted to look at the spectrum for cocaine. We can search that, load it up, and now you have the 3 different fragmentation spectra down here: low, mid, and high fragmentation spectra, as well as the metadata. So you can see molecular formula, the exact mass protein and molecule, the base peak for the low fragmentation spectrum, who collected the spectra, so whether it was collected at NIST or one of our collaborating labs that are providing us spectra to help update the library. It'll tell you where that spectra came from, and then synonyms as well, which can help in the search piece as well. This library tab on the right, the table, is all searchable. So you can do a general search up top, or you can search by name, formula, M+H, the synonyms, or the base peak. So if you wanted to say, just look at things that had 337 as the protonated molecule, you could type that in. And now you can see a list of all the different compounds in the library that have a protonated molecule at 337. In the library spectra themselves, you'll see there are some peaks that are blue and some are red. As Arun mentioned before in our curation step, one of the steps we look at is, can we explain the peaks that we're seeing based on simple bond breakages in the molecule? And so peaks that are shown in blue are peaks that we can actually explain, and you can see when you hover over them, you get a possible formula for that peak. Peaks that are in gray are things that cannot be explained by a simple bond breakage, but that doesn't mean that they're necessarily not a peak that's related to that molecule. And part of the current ongoing research is developing better algorithms to be able to assign more and more of these peaks. Just like the search functionality, we have a reporting view here. If you want to be able to print out the spectra, you can just click the reporting function and you'll get a printable version of the 30, 60, 90 volt spectrum. And I should mention that this entire DIT, it runs in whatever your default internet browser is, whether that's Chrome, Edge, Safari, all those functionalities. So all the functionalities that are typical in your Internet Explorer or internet browser, we can use here. So we can just right click and print the spectra to PDF or to the printer if we want. Zoom functionality works, Control F, all those types of things are all usable in this software as well. And you can also run multiple instances of the software simultaneously if you're interested in that. So it's designed to be vendor agnostic, so we can upload any type of text, CSV, or a JSP file, which is a JUL specific file. What we found with our collaborating labs, one of them was running a Waters QDA mass spectrometer. And the way that it exports the data is it exports all the fragmentation spectra into one Excel sheet. And so we have a converter specifically for that type of mass spectrometer. And so with this one, you'd have to upload just a generic Excel file that is the export all the different fragmentation spectra in a single Excel sheet. So we can upload this example of stenozazole. We can convert it, and what it'll do is it'll parse it into individual files, which we can then go back to our search software and we can load those files up now. So if we go back here, we now see that we have the 4 different fragmentation spectra parsed out. We can upload one or multiple of those, and again, just do a search. And I should have set this to integer mass resolution because the QDA is a single quad mass spectrometer, but we can update that, update the search, and now we get the hits, and our top one is our compound event. So that's pretty much how the DIT works. Included in the vendor or in the software download is the user manual. So we have a short user manual that you can go through, and it goes through all the different things that you can do with the software. So it gives you a summary of what the DIT is meant to do, how to download it, how to navigate it, and then how to use each individual part, the search tool, the library viewer, and the converter. And we've tried to keep this short and simple so that it's as usable as humanly possible. And so that's where we're at with the search software. Our goal is to continually update the software. Right now it's freely available from the NIST data repository site, which is down here either through that link or through the QR code. You can scan it and go there and down. And our goal over the next year or two is to add additional functionality to that. And so some of the functionality that we're working on for that process, first and foremost, is we're working to incorporate a negative mode library as well. So you could search both positive and negative ionization spectra if you wanted. We have the database builder that Arun alluded to at the beginning of the talk. It's still in a spaghetti script form. So we're trying to make it into a usable tool that laboratories could use to add spectra, create their own libraries, curate or evaluate their own libraries as well, and then add those libraries right into the DIT directly. We're working on batch processing functionality as well, which feeds into data mining capabilities as well. So if you identify a compound 2 months down the road, having the ability to go back and search the last 2 months of data to see what other spectrum might have actually had that compound in it is another functionality we're working on building in. And then the final functionality is tools for unknown identification. Arun's done a lot of work in that space. That's what the fentanyl classifier is kind of built off of, which is the figure on the right here is showing is kind of how that functionality works. And we're going to try to bring that type of functionality into the DIT for DART data as well. And as we mentioned at the beginning of the talk, we have the data interpretation tool, which was the main focus, but we also have a number of other resources that we have available to the community that we just wanted to make you guys aware of in the last few minutes of our talk here. And so all of our resources, we've been focusing our efforts on putting on a single landing page, which we're just scrolling through here, and the link is on the bottom right. But it includes everything that we've developed around DART mass spec or ambient mass spec as it relates to forensic chemistry applications. So the DIT, links to the DIT are up there, links to the mass spectral DIT, the DART mass spec libraries are up there. Any publications we've had in that area are up there, the review articles are up there. And another big thing that we've been working on over the last year or so is the development of a validation package or a validation and implementation package to help laboratories overcome barriers for implementation of this type of technology into their laboratories. And so that validation package is also up on that website. And really what it is, it's a series of documents that have base SOPs, base validation plans, maintenance plans, and the search softwares and algorithms that you would use to for that capability. And we developed this in collaboration with the Maryland State Police as we helped them bring their DART mass spectrometer systems online in their laboratory system. And so we helped them generate standard operating procedures, validation plans, how to work up all the data from the validation plans, all those types of documents. And we've made them all freely available to the community on our website down here at the bottom. We've since given that to a number of other laboratories that have just started the process of implementing DART mass spec into their laboratories. And the feedback we've gotten is having these types of documentation, as well as training. We're always open to offering training, whether it's web-based or in person, on how to use DART, just fundamentals of DART, all those types of things. But that whole process and collaboration can help reduce the time of implementation and validation by up to a year. And so we just want to make you guys aware of laboratories that might be interested in pursuing either DART mass spec or other ambionization type technologies. This type of tool is available for you, and we're always available for consultation or training and all those types of things as well. And so with that, we thank you for paying attention for our release of the DART-MS data interpretation tool. Again, the link to downloading that tool is down here at the bottom. And if you have any questions for either Arun or I, please feel free to reach out to us via email at dartdata@nist.gov. Thanks.
All right, let's go ahead and get into some questions. Thank you, Ed and Arun, for that fantastic and informative presentation. Right off the bat, we'll start with one related to kind of access to the tool. Most forensic labs cannot connect to the internet. Is there a plan to release a standalone version?
Yeah, so that is probably just something that got mixed up in the talk. It is a standalone version. It just runs through an internet browser. So you don't need to be connected to the internet to use it, but you need to have, whether it's Chrome, Edge, any type of internet browser downloaded on your computer in order to run it.
And most operating systems will have a default browser, so there isn't going to have to be any additional downloads.
Awesome. Great. Well, then let's go ahead and move on to some ones more related to the inner workings of it. So I have a question here. What is the stringency of the search parameters for ILSA?
Yeah, so there's a couple of parameters associated with ILSA. So I guess and you know, we probably didn't do a great job in this particular talk as an overview of the DIT, but the ILSA is a three-step search process. So in the first step, targets or MZ values from the mixture, from the low fragmentation mixture spectrum are identified as potential targets that we want to match. So there's a parameter there, which is the threshold intensity, above which we call those peaks targets. Then we also have parameters associated with mass tolerance. So each of those targets that we've identified, the MZ values, we look through a list of our library compounds to find matching MZ values in our library within an error tolerance. And then once we have matching library compounds to those targets, we score each of those matches based on two separate scoring algorithms, one being what we call the fraction of peak intensity explained. So that's the fraction of the peak intensity in the library spectrum that's explained by matching peaks in the mixture. And then the other and more common technique is reverse match factor, which is similar to a cosine similarity, but it's asymmetrical. So you're only computing a score with the matching peaks from the library, peaks in the mixture that are matching with the library peaks. We're still exploring which of those two methods is better for different applications. Our hypothesis is that if you have mixtures with very few compounds, either or would be great. If you have mixtures with a lot of components, then the reverse match factor, where intensity matters in the score, will be maybe a little bit worse, but we're not sure. So that's the future research that we're doing. But yeah, so those are basically the outline parameters. And in the DIT, each of those parameters is either a slider or actually, I think they're all sliders. And so they can be experimented with as the user is interested in.
Awesome. And I've got a follow-up question related to that. And I apologize if this might have been answered in that answer. You know, this is well beyond my scope of knowledge. So bear with me on this one. But how does the ILSA optimize reverse spectral searches for identifying multiple components in a mixture?
Yeah, so it's a similar kind of question, which is it's that initial stage where we use the low voltage spectrum to identify potential number of targets, and that allows us to then compute scores. Compute scores similar to a reverse. So the difference between a reverse search and what we do is that we have those initial stages that allow us to first streamline the compounds from the library that we're comparing against the mixture, and then similarly, and then the other difference is the reverse search by name kind of uses only a match factor, whereas because we're looking at multi-component systems, we thought we should explore other similarity scores like the FPI, and we're also actually working on other similarity scores here at NIST that I guess I won't go into too much detail with, but yeah, we're trying to figure out better ways to compute similarity between pure compounds and mixture components, which is of course slightly different than pure compound versus pure compound.
Awesome, thank you. We'll start with, because we're getting a couple of different questions coming in related to future tools for identification and classification. So if you don't mind expanding on that, then I think we can roll right into a couple of these specific questions related to specific uses, or future uses, I should say.
Sure. So right now our identification tool is really the ILSA, which is identifying mixture components, assuming that they are assuming that the pure compound is in our library. There's obviously going to be situations where our library doesn't grow fast enough, and so there are components in the mixture that aren't in our pure compound. We have in the EI mass spec world, we've been working on several interesting tools. For example, you may have heard, or people on the call may have heard of the hybrid search, which allows for an open modification as you're computing your match factors or computing your similarity scores. We showed a screen capture of what we call mass spectral similarity mapping. Again, that's from the EI world. But the idea is that you can use your hybrid match factors and find all the nearest neighbor compounds in your library, and then from that infer the structure of your unknown. So we want to include those types of tools with DART. Of course, it's not going to be an exact fit because the EI mass spectra are significantly more reproducible. And also those are usually collected after some type of chromatography, and so they're more likely to be pure compounds. Our spectra are more likely to be mixtures. Even the pure compounds are likely to have additional peaks that are just from from ambience as an ambient ionization method. But those are things that we're trying to include, which is basically the ability to identify things that aren't in our library, knowing that, especially in the drug world, new drugs are released well before we can add them to our library.
So in that same line of growing out the library, but maybe not so much in Steve's drugs, I have a couple of questions here, actually two in particular that are on opposite ends of what I'd say is human consumption spectrum. So the first one I have here: does the library contain any compounds related to pesticide detection?
We don't have any pesticides in there specifically, so I would say probably not at this state. The library right now has really been focused on illicit drugs, common cutting agents, common adulterants, diluents, things that you would expect to find in those mixtures specifically.
Makes sense, absolutely. The question I do have here, though, is are there any plans to add a library for food additives and food safety compounds?
That's not something we've specifically discussed, but it's certainly something we could look into. Another thing to mention is one of the big pushes for the next 6 to 12 months is to take that database builder technology or script that Arun's written and turn that into a usable type of tool, just like the DIT, so that you'd have another tab in the DIT that says, let's build a database, and it'll walk you through the whole database building and evaluation process that we're using. And so even if we don't get to a food additive or food safety compound database or a different application space, or we're slower to get to it than other people would want, we're trying to build that functionality out. That way, those types of sectors in the community would have the capability to go through, build their own libraries, build their own databases, evaluate them using the same types of evaluation steps we're using, and then have that database ready for them to be able to use the DIT.
Awesome. So really kind of lay the groundwork to have an infrastructure that can be adaptable to more specific and niche applications than what y'all are able to do just on your own.
Exactly.
Awesome. Shifting gears kind of back to the overall usage of the DIT. have a question here about defending this technique in court. Has that been done yet? Has it been defended in court? And is it considered generally accepted?
So DART mass spec has been defended in court in multiple states, I believe. So we're not forensic scientists. We're research chemists and mathematical statisticians. So in terms of the generally accepted side of it, I don't think either one of us would be the appropriate person to answer that question. There's probably other people on the webinar that would be much more knowledgeable in that space.
Sure, absolutely, absolutely. Back to questions about the library. Does it include drug metabolites or just the parent drugs? You may have touched on this. I can't quite remember if we hit that.
We didn't. At this point, it really just focuses on parent drugs because the initial application thought was seize drugs. We have been talking about whether or not to either include metabolites or have a second database that is metabolite focused. So that's definitely an area we're interested in. If there is significant interest in it, it's definitely an area that we could easily add on to.
And I'd just like to add one thing to Ed's point. To yes, it's an area we're interested in, and if other people are interested in it, they should definitely send us an email, and kind of we can get that ball rolling. It'd be nice to collaborate with some people on that.
Awesome. Well, to kind of related to that, can users contribute spectra to be included in the NIST library?
Yes, absolutely. You'll see in the current library we have there are a number of other spectra from Virginia Department of Forensic Science, the CBP labs, Maryland State Police. So we have to caveat that you are more than welcome to send us spectra. And if you want the parameters we use to collect the data, we want to make sure that the parameters are all relatively similar. So we're using the same fragmentation, voltages, all that kind of stuff. So if it matches, you're more than happy to send us the data. Just because you send to us doesn't mean with certainty we're going to throw it in the library or the database. We're going to go through our evaluation process, make sure it's of a high enough quality to include, and as long as it is, we'll put it in. And then if you want to be credited as the contributor, we will certainly do that. And if you prefer to remain anonymous, we can also accommodate that.
Perfect timing, because I think we just had a question come in from the audience asking about that specific process. So I'm glad we took care of that. Getting back into, again, kind of the inner workings of the tool, can you share more details about the mixture analysis algorithm?
Sure. I mean, so it's a three-step process, as I noted earlier, where the first step is to identify targets in the mixture, which is, of course, maybe the hardest step. We make an assumption there, and the assumption being that those peaks, the major ions in that low voltage or in that low fragmentation mixture spectrum represent either protonated molecules or base peaks from pure compound mass spectra. And so we filter first by that target, or we first filter by relative intensity to identify potential targets. We then find, based on the assumption of protonated molecule or base peak, we find matching masses in our library. And those are our potential matches for each target. And then for each of those potential matches, we compute a score, a similarity score. That also actually, it includes other, I had mentioned, the fraction of peak intensity explained and the reverse match factor. But there's also a couple other things that we consider, like, for example, we know that our matching peaks have to be within a mass tolerance. And we also know that if they're in the same direction, we call that mass bias. That's a good thing, that any mass errors are at least in the same direction versus mass errors in all wild directions. And so that's basically the ILSA in a nutshell. Also, there we should say, and we did have the paper cited here. There's a paper in JASMIS, the Journal of the American Society for Mass Spec, where we outlined the ILSA in more detail, and actually have a couple of examples in the supplementary material of how we actually calculated it, like hand calculations. I've found in the past that people seem to like reading through those detailed calculations.
And you can find that paper on the landing page as well.
Oh, right. Yeah.
Awesome. And we'll, just for the audience to know, we will post a link to that a little bit later on as we're closing up. So just hang tight. We'll make sure we get that URL to the landing page in the chat before the webinar is over. Another question I have here: what are the steps needed to expand this tool into other AI MS techniques?
Yeah, so as I think we alluded to in the webinar, just because it's called the DART-MS data interpretation tool, it really isn't meant necessarily just for DART ionization. It really can be applied to any ambionization tool. And really what it is, it's an in-source CID mass spec interpretation tool. So if you were using an ASAP probe or a secret source or a DESI source, you could still use this data interpretation tool with the caveat that you have an appropriate database or library to go along with it. A lot of other plasma-based techniques probably produce similar mass spectra to DART mass spec, and so you probably could get away with using the DART database for ASAPs or DSA sources, but for other types of sources, you may need to look at different, building your own database and things like that.
Yeah.
So going back to ILSA, is there a upper limit on the number of mixture components that ILSA can differentiate and identify?
You know, that's a great question. So we've demonstrated it with so in our manuscript with controlled substances, we've demonstrated effectively identifying 4 compounds, and then we've recently been doing an evaluation with real seized drug evidence, and that contains 5, 6, 7 compounds. There is going to be an upper limit. We're not sure exactly what that upper limit is yet, and it's a matter of continuing to experiment. Because what will end up happening is the more compounds you have, you'll end up with smaller or lower intensity peaks that just don't make it above your threshold. And so if we are able to identify those, we won't be able to identify them with as much confidence as we can identify, for example, a base peak in our mixture spectrum.
Awesome, thank you. Getting kind of back into the future for this tool, can you expand on the future tools for identification and classification?
Yeah, so the big thing that we're looking at is the hybrid search has been incredibly powerful for electron ionization mass spectrometry, and it's been people have been using it, and it's been really helpful in terms of identifying unknowns. And so implementing that same conceptual, so the implementation will be different because the DART mass spectra are different than electron ionization mass spectra, but implementing that same idea where we can allow for peaks to shift is kind of what our next future goal is. And that allows us to then expand into things like mass spectral similarity mapping, which you can then use any type of unsupervised technique or a clustering technique to try and infer identities of structures from neighboring or similar compounds. I will say, actually, the other advantage to DART mass spec, though, is we're almost always guaranteed guaranteed is a strong word. We almost always see a protonated molecule, which allows us to more easily identify or more easily predict the mass of a compound. And so we can more easily compute what's called, and this is probably a little bit too much technical detail, but we can easily compute what's called delta mass, which is the shift that's allowed in our open modification search. And so, yeah, so that's kind of the next direct step is implementing the hybrid search functionality in our DART mass spec tool, and then building upon that with different types of clustering and unsupervised learning techniques.
And the other big thing, in addition to the database builder, is building in a negative mode library.
Yes.
There's been we've increasingly shown that there is utility in negative mode, especially if your goal is more of a forensic intelligence application where you're interested in all the cutting agents and diluents and everything else that might be in there. And because of competitive ionization that can occur in some of these complex mixtures, the ability to look at both positive and negative ions helps you get a more complete picture of the material you're looking at. And so making sure that having that type of database and having the ability to do a negative and positive ion search is the other big area that we're looking to build out in the next 6 to 12 months.
Awesome, thank you. So again, in the same line of just the future for this effort and this tool in particular, and you may not be able to answer this with too much detail, but do you have any idea or what's expected for NIST's support for the tool for definitely the immediate future, and if possible, speak on the longer term future?
Well, that's an interesting question. It's a good question. That is a common challenge. Lack of funding will prevent people from I mean, we made a joke about how it's hard to procure compounds while they're relevant. Procuring compounds in general is difficult with the lack of funding. We do have, at least in the short term, the green light here at NIST to continue building the library. And that's also one of the reasons why we're trying to publish our database building software and make this more of a bigger community effort. So not just a NIST effort, because in terms of long-term sustainability, it may make sense for other people to contribute Spectra and build their own libraries and share their libraries and things like that.
And actually, and that's you brought up something I was going to mention, was when we referred to that concept of building it as a framework, as a way to expand and really get it not only adopted for use from the community, but adopted to be updated and maintained and continued on and really get a chance to grow well beyond just what NIST is able to do. So I'm glad that you touched on that, because I think that speaks volumes to what you both have been able to do in creating this flexibility and something that can kind of build its own sustainability, for lack of a better term.
Yeah.
So I got another couple questions here. We still have some time. What are some steps that are being taken to obtain large-scale data sets for the effectiveness of DART-MS? And are those data sets being obtained from labs who have a DART-MS as a validated method for drug analysis?
I'm not aware of any large-scale efforts to obtain data sets. We have been, one of the other avenues we've been talking about pursuing in the next year is establishing kind of a either inter-lab study or something like that, round robin type analysis, to start to try to obtain some of these data sets and evaluate things like reproducibility of spectra across multiple laboratories under different environmental conditions, different aged instruments, all those types of things. How is that going to affect the signals you'll get, the spectra you get, the fragmentation you all see? So that's stuff that we're working on. It's going to be in the pipeline in the near future, I would say. But in terms of other people looking to obtain those large scale data sets across multiple labs, I'm not really aware of any efforts.
You might be able to give some insight into this, or it may just be something that's a little too early to tell, but do you have any plans that you can speak of about working directly with other agencies in this effort?
We're always open to working with other agencies. If you wanted to be more into the R&D and the development of the software, we certainly would welcome you to kind of join the team of collaborators that kind of get the early access to the software as we develop the functionality, kind of as a beta tester, providing input on what works, what doesn't work. We're certainly open to that. Just send us an email at dartdata@nist.gov and we'll get you on that loop to be included in those types of discussions, those types of feature building. And then also any other questions that you might have or any other types of research that you might be needing in this kind of realm, definitely reach out as well. We work with a lot of labs across the country and internationally. So we're always happy to expand our network.
Awesome. Circling back real quick, just kind of, again, getting back into what the library includes or doesn't include. Can you confirm, does the library include common drug cutting agents, dilutants, and other components that might be found in the drug seizures that can be used for drug intelligence or linking seizures?
Yeah, that's actually one of the big drivers for what kind of spawned all this work, was the need or the desire to move towards a forensic intelligence association signature, whatever you want to call it. So we really have focused on making sure a lot of those cutting agents and diluents and compounds are in the database. A lot of them also, I should mention, a fair number of them are not responsive either by DART or only responsive in negative IMO, which is a bigger driver for us to move into that negative ionization capability as well. But those types of compounds are definitely in there. And if there are compounds that are missing that are important, you can always let us know. If you have the spectra, send the spectra. If you don't have the spectra, let us know what those compounds are, and we'll try to include them in the next. We release roughly quarterly updates, and so we can make sure that they're included in the next update.
Awesome. I got one more question here on our list, and I'll go ahead and put this out to the audience. If you have any more questions, please feel free to send them in. We still have a little bit of time, but this might be a good one to wrap up on, because it kind of ties back into Well, I'm hoping that maybe y'all both can get a little more personal in your journey and getting to this point with working in this field, specifically related to this tool and this effort. We have an audience member who is a criminology student who's interested in addiction and drug use and looking for advice. And maybe more so, I was thinking if you have any advice or anything that kind of is a helpful perspective on on the path to getting to this point, or how y'all got here.
Yeah, you can go for it.
I'm trying to think what my best advice would be. The biggest thing that we found through this whole process, and the most rewarding piece of it, is making sure you work with whoever the end user is. So in our case, it's the drug chemists and the practitioners on the bench that have to go in, day out, and do this type of analysis. And so making sure that you always keep that in mind is what they need, what are the challenges they're facing every day, what are the ways we can help make the burden a little less, make the backlog a little lower. Always keeping that in mind has been the biggest thing. Never being afraid to reach out and just ask a question or ask people to participate in studies. I mean, we've done studies in the past where we originally put it out into the world and everybody that we asked to help out with said no. And then a couple of years later, everybody was saying yes, and people were asking us if they could join in on the study. So keep your head up, just push forward. And
yeah, I think even, beyond, forensics and whatnot, I mean, I think Ed's advice of building a network, and I know it's common like LinkedIn talk, but it's true. It's good to know people, it's good to meet people. You'll be surprised, when I got into forensics, I think we kind of mentioned it, I didn't really do forensics up until I really met Ed. And it's been exciting to meet and talk to people and learn what their actual problems are and focus our research a little bit. I feel like it's been an incredibly productive year because of that, because we've been able to meet with end users and focus in on real, solid problems. And also the other thing, I mean, with the student in particular, so at NIST, we do have programs in place, research programs. If the student is interested in doing research related to, I think you had mentioned addiction and drugs. We don't really do that type of research, but, chemical identification of drugs or things like that. We've got research programs specifically for undergraduate and graduate students, as well as postdocs. And so if they're interested in doing that type of work, and if they're not interested in doing that with NIST, I'm sure a lot of other organizations will have these types of programs. And so it's good to get in early and get your name in there and let people know that you're interested in working.
Absolutely.
There's definitely, and there's definitely no shortage of job opportunities. It's just there is a shortage of labor, and so get your name out there. People will try to find you.
Awesome. Great advice from both. I mean, it really is, asking questions of people who are doing the things that interest you, that you want to work in, I think is definitely advice applicable across the board for any students. So I really appreciate y'all's both giving your insight into that. That is it for our time. We have run out of time, unfortunately, so we'll go ahead and start closing things up and saying thank you to both of you for presenting and taking the time out of your day to provide the material and the questions and all that. It really does mean a lot to us and our audience. So Dr. Cisco and Dr. Morthy, Ed and Arun, thank you again for taking that time to provide this wonderful presentation.
Thank you.
No problem. Thank you.
And then for the audience, like I mentioned earlier, we're posting that URL to the landing page in the chat. Just note that the chat system does not allow for direct hyperlinking. So if you click on that URL in the chat, it's not going to take you anywhere. You'll need to copy it out of the chat and then paste that URL into your browser's address bar in order to actually navigate to the DART-MS DIT landing page. And here is that URL as well for anybody that would like to type it in that way, and contact information for Ed and Arun. Before you leave today's webinar, we ask if you could participate in a short 5-minute survey. You will see a checkbox icon directly above the chat simply click that button to be linked to the audience feedback survey for today's event. Your feedback about our webinars is critical information that helps us improve and continue to provide you the best webinar experience possible. If you could please take those few moments to complete the survey, we would greatly appreciate it. Lastly, across the top of the live stream window are links to all our social media profiles, including Twitter, LinkedIn, and Facebook, a link to our website at forensiccoe.org, and a link to listen to episodes of the FTCOE's podcast, Just Science. There's also a link to sign up for the FTCOE newsletter so you can stay up to date with everything that FTCOE is doing in support of the forensic community. Thank you again for attending, and we hope everyone has a great day.